I think my talk marks the transition to CRISPR-related talks for the rest of the session. And I'll present EpiCRISPR, a new method which uses epigenetics data, prediction based on high-throughput data and flanking sequences to improve endogenous on-target efficiency prediction. This work was done by my former master's student, Michal Archimi. So just a short biological introduction. I think most of the crowd here in the audience know about CRISPR Cas9 genome editing. A programmable 20 nucleotide guide RNA leads a Cas9 enzyme to a complementary sequence in the DNA, in the genome, which must be followed by a PAM sequence, which in the case of a Cas9 is NGG, and then it creates a double-stranded cut. Once a double-stranded cut is created, a cellular repair mechanism comes into action. And in most cases, we don't have a repair DNA template, so we get a repair by non-homologous end joining, which leaves a mark that could be an insertion or deletion, which we can then identify in an experiment and measure the cleavage efficiency. The goals in the field, kind of two very major goals, is first to have high on-target efficiency, meaning we want to design a guide RNA that will cut the gene that we want to cleave or edit with high efficiency. And we are also concerned by off-targets, meaning editing at sites that we didn't intend to edit. In this talk today, I'll only focus on the first goal, which is achieving a guide RNA with high on-target efficiency. I won't have time to go over the experimental assays that measure on-target efficiency, but I'll just show you the kind of output or experimental readout that computational people like me see. We see a list of guide RNAs, which you see here of the 20 nucleotides, followed by the PAM sequence. And you can also see some upstream or downstream nucleotides. And here on the right column, you see the on-target efficiency, which is the label that our model or method is going to predict. So this is both the experimental measurements and also what our method is going to predict. And there are high-throughput methods that were very successful and produce lots of data, tens of thousands of data points. But the problem with these experimental assays is they measure the on-target efficiency in a synthetic context. So most of them are based on lentivirus insertion, where we don't know exactly the genomic region where the target was inserted. On the other end, there are endogenous experiments, meaning experiments that measure the editing at the genomic location of the target site. So these are more biologically relevant, but they are much more difficult to run and perform, so we get less data from them, maybe only a few tens or hundreds of data points, but they do give us editing at the genomic context. And here is the main goal of many of the data-driven method, giving a sequence, which you see here, the guide RNA, we give it as input to a machine learning method. The machine learning method is going to predict the on-target efficiency. And there are plenty of methods that have been developed throughout the years. So what are the gaps of current methods? So there was a recent study actually from last year by Ito et al., where they tested multiple state-of-the-art methods on a data set that they created, produced 205 editing measurements of guide RNAs, and they showed that the best performance was achieved by a chop-chop of a Spearman correlation of 0.43. And one of the reasons why these methods fail to predict endogenous editing is that they don't include epigenetics data and other cellular factors which are associated with on-target efficiency. So we came to this problem and wanted to resolve this issue, and indeed to include cellular factors and epigenetics data to improve the prediction of on-target efficiency. So we started with collecting the data, and luckily there was an opportunity with the Lina et al. Dataset, which I'm showing here in the first row, which had almost 1,600 data points. So this was, I think still is, one of the largest data set of endogenous editing, and we used it for both for training and evaluation. And we used 4 additional data sets for independent evaluation. We didn't do any algorithmic development or any training on this data set, and I'll show the results later on. For encoding the input, we use the very common one-hot encoding of the guide RNA and flanking sequences, which I'll show with the length that we used. And we added the epigenetic data as scalars, so 1 or 0. If in a bed file we saw that there is the epigenetic marks or not. In case of methylation, we had some more quantitative data, so we use the average methylation status, DNA methylation, over the guide RNA, and we also use the prediction by CRISPR-on method as an additional feature. We tried different architectures, CNNs, RNNs, a combination of CNNs, RNNs, and also the multiple layered perceptron, which performed the best. And we also liked it best because the simplest has fewest parameters, so it has the advantage of reduced overfitting risk. So moving on to the results, we started with experimenting with the flanking sequence length, both downstream and upstream. And we evaluated the performance in 5-fold cross-validation on the Lina et al. Data. And here I'm showing the results of different combinations of downstream and upstream flanking sequences. So here you see in the upstream and the y-axis, I think there is almost no effect when we add nucleotides. So we decided to add 0 upstream nucleotides. But on the other end, on the x-axis, you see the downstream nucleotides, and you see that they do have an effect, that they do improve prediction performance. So we decided to add 9 nucleotides downstream of the PAM sequence. Now moving on to the effect of adding additional features, we start with the sequence-only baseline, which is at 0.375. And here I'm showing you the addition of each of the epigenetic features, and each addition of each feature independently improved prediction performance. So we're very happy to see these results. And also when we included all of them together and also with the prediction by CRISPR-on method, we saw further improvement up to almost 0.52 in Spearman correlation. So we were happy to see that additional feature help. And what was impressive is that those features were collected from other studies that we downloaded from ENCODE or for the DNA methylation atlas. They were not collected from the same study of Lina et al. Now moving on to the cross dataset results, we trained our model on the Lina et al. Data, and then we tested it on other cell types and other datasets, independent datasets that were not part of the algorithmic development or the training. So here I'm showing the results over K562 cells, and we see a huge boost in performance by adding the epigenetic features. We see the same trend with HCT116, with EC293, and also on the independent T-cell data of Ito et al. Just to remind you that here we go to a Spearman correlation of almost 0.65, and this compares to the one that was achieved by Chop-Chop of 0.43. So we're very happy to see that EpiCRISPR generalized well to other datasets, other cell types, and the trend in epigenetics contribution is preserved. Last, I will show the interpretability analysis that we performed. We wanted to ask what are the important features that the model has learned, and we used gradient descent to find the input that maximizes the output. We found some sequence preferences, and I won't have time to focus on the novel preferences that we found, but also epigenetic preferences. Some are related to active regions in the genome and other to silenced regions. So we were able to find both known and novel preferences on the sequence and epigenetics level. So now moving on to the conclusion, in the future, we'd like to include additional features and also maybe impute them by computational methods. We'd like to also test the addition of these features on CRISPR off-targets and repair outcomes. And of course, the most popular hype in the field are the language models. So to summarize, we developed EpiCRISPR, and we improved on-target prediction performance. And I think the most relevant and key point that helped us was the endogenous datasets that were available and allowed us to achieve this achievement. So with that, I thank you for the attention, the funding agencies, all the data, methods, trained models are available on GitHub, and I'm happy to take questions.
Thank you. Very nice talk. I was wondering, because all these epigenetic markers associated with R-loop formation, for R-loop formation, whether or not can actually using R-loop formation information to predict whether or not it's actually can be more direct guided.
So what formation?
R-loop.
R-loop, okay. Yeah, that's a good, we didn't try to like we can take R-loop data and see how it well correlates. Yeah, so there are maybe other epigenetic factors that are correlated with those. Yeah, that's a good idea. We can try. Yeah.
I've got a question. It might be a naive one. What's the explanation for the epigenetic marks having a different impact in the different cell lines? So it looked like they made a bigger difference in some cell lines than others. Is that because of the depth of the data set, or is there a biological reason?
So we didn't test this hypothesis. My most probable guess would say the data quality, right? Some experiments have higher quality than others, but we didn't go into the details and depth to ask that question.
Okay. Perhaps I missed this, but which type of over here. Which type of Cas9 enzyme was it? I know there's a lot of...
yeah, this was Cas9.
Was it like high fidelity, enhanced specificity, wild type?
I don't recall. I don't think they used one of the variants. I think they used the most popular Cas9, but I will have to check. I'll have to check at the Lina et al. Study and to tell you.
Yeah, entirely fair. We should also talk after.
Thanks.
Okay, if there are no further questions, then thank you very much. Thanks.